all plugged in there. So, yeah. Welcome to Patho. Again, my name is Brian. Please do not give me any titles. It makes me feel uncomfortable. I am not a responsible person, so just get that out of the way. So, yeah, Mondays, we're going to be meeting in person. We'll be in here. Class starts at 8:30, but we'll talk about like how quizzes work and stuff like that. If you guys have any questions, just feel free to interject. Just that way I can address it there. Okay. All right. So just starting off with the admin stuff, this is going to be really quick, and then we'll jump into lecture material here. So standard stuff you've probably heard before in any of your other classes. So that's me right there. That's my email. That's my phone number. That is my actual phone number. Feel free to write that down. Feel free to text me. That's fine. I've had students text me 8 p.m. on Sunday. Can't guarantee I'll respond to you, but that is a way to get a hold of me, just in case like you have a burning question, your email is not answered. Okay. Please let me know who's texting me though. If someone just randomly asks me, hey, what are we doing tomorrow? I have no idea. Okay. So just let me know, like, hey, this is XYZ. What's going on? Okay. Yeah. So my area of expertise is more biomechanics, so physics of movement and kind of stuff like that. So if you have any curiosities, let me know. If not, most nursing students don't. Syllabus approval. So on the website, and I'll show you where it's at, you have to consent to the syllabus. It says it's a quiz, but I'm not asking you, like, it's not a full-on quiz about the syllabus. It's saying, do you consent to the rules laid out by the syllabus? You got to make sure you get that filled out and submitted within about 24 hours. The sooner you do it, the better, because I can't post your grades without your consent. If you don't agree with the syllabus, then the syllabus doesn't really apply. So if you're a person that has anxiety, you're going to see zeros all the way through your grade until you get it approved. So just knock it out sooner rather than later. It's like 14 questions. Do you agree? Do you understand? Do you have any comments? That kind of stuff. Okay.
It's on Canvas.
Yeah, and I'll show you guys where all the link is. I'll show you guys the Canvas homepage and where to find all of the materials. So this is kind of how things are supposed to work. Day one, we're going to meet in person. We'll be here 3 hours. Some days we get out early. Some days kind of goes all the way to the end. Just to give you full heads up. We will take breaks in between though. I'm not going to make you sit here for 3 hours straight just listening to me droll on. So we will take breaks. Day two will be the online day. I'll show you on Canvas as well. I have my Zoom link. You guys are just going to jump into there. And then basically this format carries over from the online field. You're going to see my face in a little square. You're going to see the screen. I'm going to be writing on the screen. So very similar. Deadline wise, the only deadline really right now is the syllabus review. Okay, there's no projects, there's no papers, there's no presentations, no group projects for this class. It's straight up just like undergraduate. Quizzes, exams, that's it. Okay. Speaking about quizzes and exams, Patho is a hard class, guys. That being said, every time I see you, you're going to have a quiz or an exam. That includes tomorrow. This is the only day you will not have a quiz or an exam. So just heads up. Okay. I promise, I know it's a little anxiety. I'll walk you guys through what a quiz looks like. When it comes time for the exam, I'll give you guys plenty of examples. I ask a lot of questions throughout lecture. So one of the things I recommend to students, first pass here, when you guys are sitting here, just listen, write down any example questions I give out in lecture. You might not have the same exact question on the exam, but you might have something similar. Then you have the whole YouTube recording, turn it on 1.5 play speed, so it's a little bit faster, and then write your notes. Just use this as a first pass to kind of get an overview, because some of these lectures, there's a lot of details, okay? So quizzes will happen technically at the very first 15 minutes of class. So class starts at 8:30, you have till 8:45 to complete your quiz. Quizzes are all multiple choice, 13 questions, okay? Now, I don't expect you guys to be in your seat at 8:30 taking your quiz. I try to figure it out a little bit. I will have the quizzes, and I'll write all this down for you guys. I will have the quizzes open up at 7 in the morning on Mondays. So You could take your quiz at home, then use that time to commute here. I don't want anyone to, I hate commuting. I take the bus. I don't want you to feel like you're rushing, trying to get to campus at a set time. So if you want, quizzes are open at 7, you can take the quiz at home, or you can take it here. Either one's fine. So even though class starts at 8:30, I won't start lecturing till 8:45. If you want to take your quiz here, that's totally fine. The advantage of that, you can ask me questions as you're going through the quiz. Like, what did you mean by this? Or is this a typo? Either option's available to you, okay? The quizzes and the exams are closed notes. So don't have your notes in front of you, whether you be at home or here, okay? So quizzes, there's going to be 15 total throughout the entire quarter. The lowest one gets dropped. So let's just say you knock tomorrow's quiz out of the park, it's 100%. Next one, 40%. Don't worry, that 40% gets dropped. The lowest one always gets dropped. It's already configured in the grade book. So when you guys look at your grades on Canvas, that's the most up-to-date grade. Okay. There might be some select alls in there. Sorry. For whatever reason, the NCLEX likes select alls. So select alls is like, which one of these apply to this question? A, B, D, or something like that. That's a select all. Okay. I try not to have too many in there, but those are a thing. The quizzes and the exams are set up like your HESIs and your NCLEX. So your HESIs are the big exams at the end of each nursing class. So in health assessment, you're going to have a HESI for health assessment. The HESI are small milestone steps all the way to the NCLEX. So if you've taken a bunch of HESIs, you should be ready for your NCLEX. That's the idea there. How your HESIs and your NCLEX are structured, one question at a time. So you can't scroll through the quiz, you can't scroll through the exam. You got one question, you make your option, you hit next, and you move on. There's no backtracking, there's no changing your answers. Again, you can't go through the exam. To help take the edge off, your quizzes and your exams are structured the same way. Okay, so one question at a time, just choose your option, hit next. There are 6 exams in total, so 5 unit exams, 1 final exam. It varies depending on how spicy I'm feeling writing your guys' exams. Give or take, it's about 40-ish questions. Most of them will be multiple choice. So if it's a 40-question exam, you're looking at maybe 38, 37 multiple choice, maybe a couple select alls in there. There are short answers like fill in the blank, where you'll actually type in your response. And then there are like essay questions where you'll actually write out. Don't write me an essay. This is not English. I suck at English. You're going to find out my spelling sucks. Bullet lists. Like, what are the 6 things that define cancer? That kind of thing for an essay question. Okay. Final, a little bit longer. We'll talk about the final when we get there. Final is 50% current material, which will be hormones, endocrine stuff, and then 50% cumulative. So heads up, please don't forget, don't learn and dump. It's really easy to do that in nursing school. Totally hear you. I've been through it. And I try to like keep bringing up older material in lectures as we move on, but try not to just learn something and then dump it. Okay. Questions on quizzes or exams. So, and I promise to write down all of the quiz times, the windows that you'll have open. Again, if you want to take it at home, you're more than welcome. You want to take it here, that's fine. I'm usually here by, I want to say 7:30. I try to be here by 7. Try. It's hard. It's an hour commute for me. So I live in Broomfield now, but I just trying to get my ass to the bus stop sucks, and bus people are weird. So I try. That being said, so we have class today. Tomorrow's class is online, yeah? I just recently moved. I don't have internet where I'm at. So I've already pre-recorded your lecture for tomorrow, and I'll show you where all the links are. I won't take attendance or anything like that. The way I'm taking attendance is your quiz. So jump in, I give you guys, it's like an hour window for you to take your quiz. And if you take your quiz, you're good, you're done. And then you're gonna watch that pre-recorded lecture. Tomorrow's lecture is cancer, okay? In theory, I'll have internet after tomorrow. Yeah. So that's fun. All right, so as I mentioned, this is a very straightforward class. 15 quizzes, but only 14 of them count. Each quiz is maybe worth like 1%, that 1.07. So they're not much. The quizzes are not meant to be penalizing. They're not meant to like punish you. They're to keep you up to date with the material. So if you have something going on and you drop the ball 1 week, that's fine, you can get dropped. Okay, but ideally the quizzes just make sure that you're kind of keeping up with the material as we go along, so you're not cramming for each exam. I'll tell you right now, with each lecture being give or take 3 hours, and you have 4 lectures per exam, that's 12 hours worth of lecture. Do not try to cram that. That will hurt. Okay, so again, the quizzes are not super difficult. They're pretty superficial. If you're just paying attention, you're listening, you're answering the questions as we go along, you'll be in good shape for the quiz. Okay. But yeah, quizzes, exams, that's it. Ad drop deadline is gonna be October 10th. And the withdrawal deadline is November 14th. Okay, so this will play a little bit more later, but basically the withdrawal deadline, that's after exam 2. We'll talk a little bit more about that as we get a little bit closer. Okay. Also, I can't see what you guys see, so if you need me to scoot it up because my fat head is blocking it, let me know. Okay. So at DCN, at this school here, you need a 78% to pass your class. Okay, so that's the cutoff. Anything lower would not be considered passing. Now, a little bit of a horror story here because I'm trying to put the fear of God in you. If somebody ends up with a 77.99 at the end of the quarter, what do you think happens? Yeah, this is considered failing. There is no leeway. There is no leeway, even 0.01. This is where your quizzes really can make the difference there. Try to take your quizzes seriously because you don't know when you need that extra 1% to sway you to the right direction. Okay, so just really trying to reinforce that. There is no rounding. When we calculate grades in the grade book, it actually calculates out to the thousands. This is the only time that rounding would quote unquote apply. So if your grade is 99.4, that would be failing. 99.5, that would be passing. Now, don't ride the razor's edge. Aim higher. Okay, just shoot higher. Just heads up about that. It's a very strict policy. You can take it up with me, but I'm going to be honest with you. I'm so low on the pay grade scale that I can't do anything about it. Okay. All right, so quizzes. Let's say, for example, I'll say Rivka.
Yeah.
Okay, Rivka, let's just say you have a dog and your dog's in the emergency room vomiting. You can't make it to class. It's okay. Life happens. I'm not going to be like, why are you not prioritizing your school over your dog or your family? I don't know. Life happens. Okay. I try to be flexible with you guys because I want you to be flexible with me. I'm not perfect. Okay. If something comes up, email me, text me, let me know, and I will work around it because we can get you to take the quiz at a different time. Exams are a little bit harder to navigate through, but again, the sooner you let me know or even earlier, the better. Okay. All I'm saying is work with me and I'll work with you. Okay. Yeah, then there's like this 10% deduction for unexcused stuff. So take, for example, if you don't take your quiz, you're like, can I take my quiz? I'm like, well, why didn't you take it in the first place? Oh, it's because I slept in. I gotta take 10% off. Okay, so that sucks. Don't do that. All right, so syllabus approval, there's that. Let me show you guys the course website. So this is what happens when you log into Canvas. This is that homepage right there. There's my info again. Okay, office hours. My schedule is stupid crazy. That's why I don't have any set office hours. It doesn't mean that I'm not willing to meet with you. I am more than happy to meet with you. I straight up met up with students, I think Saturday and Sunday last quarter. Everyone's schedule is crazy. If you want to set up office hours, you want to go over material. Do you want to prepare for a quiz? Do you want to prepare for an exam? Do you have questions? Do you need me to re-explain stuff? Shoot me an email. Let me know what days and times you're free. Give me like 3 options and I'll get back to you and we'll set something up. Either we'll meet here in person or via Zoom online, whatever's easier. I have the ability to draw and communicate online, so that's why I don't mind doing online. Okay. So there's the classroom D, that's us right now. That's the link to my Zoom room for Tuesday. Okay, again, because I'm in the process of moving, no lecture, no official lecture tomorrow, just that pre-recorded lecture. Okay. I try not to do pre-recorded lectures. I don't want to turn this into a YouTube university. That just, unfortunately, the timing of it sucks. So the other tab, does everyone have their computer-ish? Pull up your Canvas right now. I'm kind of curious about something. And if you can't do it right now, don't worry about it. But if you pull up your Canvas and you log in and you go to your settings, what time zone are you guys set to?
Pacific.
Change that to Mountain Time, because otherwise you're going to freak out and think that you missed an exam 2 hours ago. So there should be like an edit button at the bottom, I think, for you to, mine's a little different, but there should be like an edit button at the bottom for you guys to change your time zones to. It's on the side. Perfect. Oh, thank you. Yeah, the edit settings right there. So change that to Mountain Time just to save you some headache. That will apply for health assessment too, and here on out here. So you don't need to do it once. Okay. So next up, modules. Modules is where you guys are going to be pretty much living. Here's your syllabus review form right here. That's what you're going to click. That's what you're going to start submitting. That's what you need to approve to get it approved or whatever. Okay. So it's right in the modules there. When I record a lecture, I'm going to post it right here. It's just a link. You click on that, it'll pop up a YouTube video for you. So you don't need to do any copying or pasting or anything like that. If you want to take notes on your computer, here are all of the PowerPoints for the entire quarter. Okay. If you don't like 6 slides per page, you want 4, you want 2, you have that PowerPoints, you can print them out as well. Okay. I personally, I annotate everything via PDFs. So if you like PDFs, these are all the PDFs of the lectures. Same material, same thing, just different file formats. Okay. So that's where all of the quote unquote resources will be. This tab right here, your quizzes, that's where you will find your quizzes and your exams. As you can see right now, you should actually be able to see quiz 1 on your guys' end. Okay. When it comes to quizzes, so let me write here quiz times. So for you guys being cohort A, I have it set up so that way your let me rewrite this. Day 1 quizzes, they will be on Monday in between 7 in the morning and 8:45 because we start class at 8:30. Okay, that means the quiz will open up between those times. The quiz should only take you 15 minutes. The quiz is only going to take you 15 minutes. It'll log out after 15 minutes anyhow. So again, if you want to wake up early and do it, you want to wait till you get to campus to do it, you want to do it while you're on the bus, either one's fine. Okay. For day 2, so the online day, that will be Tuesdays. I have it set between 7 and I believe 8:15 because class starts at 8 on Tuesdays. Okay, again, any 15-minute window you got within that time window, you can take your quiz. Okay. Exams will happen in class, so that's why I'm not writing any exam times. And we'll talk a little bit more about that when we get a little bit closer to exam 1. Okay, it'll be in a couple of weeks. Okay, so really, yeah, modules and quizzes are really where you're going to be living. The textbook for this class. So you got to go through. Do you guys know how to access your textbooks yet? Okay. If you don't, just come see me. That's fine. I'll show you guys how to get to it. The textbook for this class, on the syllabus, there's chapters of the required reading. I don't require those. Okay. Couple of things about the textbook. I'm a teacher. I try to take pride in the fact that I want to teach. I don't want to be the librarian telling you to read chapters 12 through 15 quizzes on Monday. Not my style of teaching. So the textbook, let me be frank here. I will not have a random question from the textbook on the quiz that I never went over. Not doing that to you. The material that you are responsible for is the stuff I write down and the stuff I say out loud. That's what's going to be testable. Now, does that mean the textbook is absolutely useless? No. It's a great secondary instructor, but the reason why I don't really emphasize using the textbook, it goes into so much detail. This textbook is written at a pre-med level, or I should say med school level. It goes into way more details than I go into. Okay, do you need to worry about what the cytoplasm of somebody with leukemia looks like as a nurse? Not really. Okay, so that's why I'm saying the stuff we go over, that's testable. I'm not going to have a random question from the textbook, but your textbook can be useful. I'm not the best at explaining everything. If maybe my explanation doesn't sit well with you, go into the textbook then. Okay, but no, the required readings are not actually required. Okay. Questions so far about like how this class is going to run, stuff like that? This is a bit of an information overload. Good so far? All right. Yeah. Cool. All right, so yeah, we are going to jump into lecture material here. Do you guys want to take a little break to like meander around, get some blood going, or do you want to just jump right into the material for a bit? Democracy shrugs its shoulders. All right, we'll just do a little bit of cell bio stuff. You guys have heard this since high school, so it's not crazy new information. Okay. All right, so cell bio and genetics. Cool. So also, if I just skip past a slide, like take for example this slide here, do you think I'm really going to test you? What was the fourth word on slide 3? Don't worry about it. All right. So starting off here, bunch of terms. Okay. We're going to go through and define these terms, then I'll kind of give you guys some context about how I might ask questions about these terms. Okay. One thing about pathophysiology, one thing about nursing diseases, know your Latin. You're going to hear me chant this like a crazy cult member throughout the entire quarter. If you know a little bit of Latin, it's always going to give you a starting point. Okay, it's always going to give you some sort of starting point here. So take for example, let me know if I choose a color and you can't see it, okay? Organelles translates to as little organs. Little organs, i.e. these are the organs of a cell. Okay. All right, nucleus. You guys remember that one? What's the nucleus do? Brain? Stores the DNA, perfect. And that's what I try to do is like, I want to have like an organic kind of conversation with lectures. I would say if I ask a critical thinking question and you know the answer right away, just give it a second, let people marinate, don't spoil the answer for everybody, okay? But yeah, so I'm gonna say command center. You can say the brain, that's fine. Command center, and it stores DNA, okay? If you cannot read my handwriting, let me know, okay? I have arthritis in my right hand. Years of stupidity. So if you can't read it, just tell me, I'll rewrite it. Okay. Endoplasmic reticulum. This one's a little wonky. There is a smooth ER and a rough ER. Okay. So for the smooth ER, this is going to be lipid processing. Okay. Lipid, i.e. fat processing. Rough ER, this is going to be protein processing. Okay. Ribosomes. Ribosomes, there's a key word here. And notice how I kind of give you a hint. There's a key word here. Ribosomes, they translate amino acids. So heads up, I abbreviate amino acids, AA. Translate amino acids into, oh, oops, I messed that up, sorry. Yeah, not amino acids, RNA, translate RNA into proteins. Sorry. Ribosomes translate RNA into proteins. Your Golgi complex, this is going to be responsible for kind of shipping. You can do transporting if you want. That's another word here. I'm just going to say shipping materials around the cell. So Golgi complex ships materials around the cell, transports material around the cell. We're talking about proteins, secondary hormones, etc. Lysosomes, these are going to play an important role, especially when we start talking about the immune system. When we lyse something, that means to break down or cut. I'm going to say break down stuffs. Now that can be like a macrophage breaking down bacteria. It can be breaking down like old cell parts. Okay. So lysosomes, their job, it's basically like a little vat of acid. We just throw stuff into the vat of acid to dissolve it. Peroxisomes. You do not need to worry about peroxisomes. So this is an example of an artificial line in the sand I need to draw. When you guys become a nurse, if you use the term peroxisome at any point in your nursing career, I will pay you $200. Right? I mean, like how often does the peroxisome come up in practice? It doesn't. So this heads up here, you know, I think the peroxisome is freaking awesome. Does anybody like Krebs cycle, fatty acid chains? Freaking awesome. Do you need to worry about that as a nurse? You can explain that to your pediatric patient. So sometimes we will just like straight up omit stuff like that. If I draw a line through that, are you going to expect a question on the quiz or the exam? No. Okay. And that's what I'm saying, like your textbook will go into about peroxisomes. I won't. Okay. Mitochondria, what's that? What'd you call it?
The powerhouse of the cell.
You know, this is kind of an interesting one. I don't know.
Memes you see everywhere on social media.
Yep. So it is the powerhouse of the cell. My beef with it is, what the hell is a powerhouse?
It's a very general term.
I mean, but like you use it in the 90s in the gym, like, oh, that guy's a powerhouse. But like, you don't drive past the power plant like, oh, that's a nice powerhouse. I don't know how house got into it. That's just my beef. Cytoskeleton, cells, skeleton, so just the internal framework. Okay. So your quiz is going to be very superficial. It might ask you, what does a ribosome do? Something about translation. What does the mitochondria do? Okay. So quizzes, again, are meant to be very superficial. If you're here, you're awake, you're taking notes, you're answering my questions, you'll be fine. The exams are going to be a little bit more about application, and I'll give you guys some examples. Okay. So the following here are going to be metaphors. No, I will not ask you what what the postal office is a metaphor of a cellular organ. This is just a class exercise. Don't read too much into this. Okay. But for example, you guys have your list there. What would be most akin to the post office? Correct. Golgi complex. Right? Just because it's going to be sending stuff all over the cell. All right. I appreciate you sticking to your guns. All right, so this one should be pretty straightforward, yeah? The mitochondria. All right, so with this one here, I'm gonna go a little bit more and kind of elaborate upon this. Your mitochondria is the power plant, the powerhouse of the cell. What does it make? What is that? What ATP? So ATP is that energy. What does the mitochondria burn? What does the what do we burn to make this energy?
Lipids.
Lipids is going to be Krebs cycle. Mitochondria is going to be kind of it's going to be straight glucose first. Okay. So glucose here is a molecule. Does anybody remember from their phys, how do we store glucose in the body? Glycogen. Okay, so glycogen is the storage. Okay, so I usually try to do a lot of analogies when I teach as well, just to kind of help give you guys a better grasp of the material here. So talking about parking in downtown, you want to pay the meter, right? The meter gives you time. Let me say like time on the meter. How do you buy time on the meter? Money. Let's just say like quarters if we're old school. How do you store quarters? I was going to say like in a roll, right? You guys have seen those rolls of quarters? Yeah. Okay, so why do I bring this up? Rolls of quarters. If I drive to downtown and I have a bunch of quarters in my pocket, I walk into the building, what's going to happen to my car? I didn't put the quarters in the meter. They're still in my pocket. I'm going to get a ticket. I'm going to get booted. I'm going to get towed. Why? I had a bunch of quarters though. Why am I still getting towed? I didn't feed the machine. Now, if a person has a bunch of glucose, does that mean that they have a lot of energy? That's the key difference here. Just because somebody has a bunch of glucose doesn't mean that they're burning it. Glucose is not energy. ATP is the energy. Okay, so just heads up about that. Glucose is not the energy. You burn glucose to get said energy. Okay. All right. Dump truck. Yeah. Lysosomes, because we're going to use it to get rid of the old stuffs. Okay. And if you need me to go back, or if I'm going too fast, just let me know. I'll slow down. Okay. All right. The library. I agree. It's going to be in the nucleus. Actually, let's use. It's going to be the nucleus. All right. I'm going to make you guys a little deal. I want to offer something. The wager or the deal is you will not judge me on my spelling during lecture, and I will not judge you on your spelling during exams. Deal? Okay, so if you have a typo or you can't spell something correctly, as long as I know what you're talking about, you'll get the points. Okay, so just heads up, my spelling sucks. So your nucleus is going to be equivalent to the library. Okay. Now your library holds books, and inside said books you find texts. Your nucleus holds DNA. Those are your texts, or like the lines of text. I'm sorry. How do we store DNA in the nucleus? You have 23 from mom. Yeah. Chromosomes. Okay. So that's how you can kind of think of that organization there. Chromosome is just a bunch of DNA. Okay. All right, so this kind of gets into like the central dogma of cell bio here. Pay attention to how I word these questions, okay? DNA, if I undergo replication, so this is a process. If I undergo replication, what am I making? I'm making more DNA. So I can ask you a question. What is the end product of replication? Sorry, end product. Like, what do we get at the end of more DNA? What is necessary to undergo replication? We start with DNA, we make more DNA. You see what I'm saying? Like, don't overthink this here. What is the end product of replication? DNA. What do you need to undergo replication? DNA. Okay. So that's why I'm saying, like, don't overthink this here. Transcription. What is the end product of transcription? RNA. What is necessary to undergo transcription? DNA. Okay, so we're going to rewrite this out here, kind of give you some more practice with it. Okay, so I'm just rewriting what that last picture was. So DNA, if I start with DNA, more DNA, what's that process called? Okay, now if I take this DNA and I turn it into RNA, what's that process called? Transcription. And then finally, I'm going to take that RNA and turn it into a chain of amino acids or a protein. What's that process called? Translation. Yeah. Now, translation should ring an eerie bell for you. In fact, I kind of hinted at it earlier. When did translation come up in lecture? The rough ER and the ribosomes. So that's me giving you a hint. Where does translation happen? At the ribosomes, yeah. You guys want me to close the door? And we can close the back one too. It's still loud. Okay. So translation happens at the ribosomes. Good. Let me give you guys another analogy to kind of help break this down here. So I want you to imagine in the 90s, do you guys remember public libraries, how they had internet dial-up? Do you remember how it cost money to print? It costs money, like a lot, good chunk of money to print, right? So let's say you're at the library and you find a nice online recipe for beef Wellington. Okay, so you find a nice online recipe. Now there are plenty of recipes for beef Wellington on the internet, yes? Okay, now you don't have money to print it. So what are you going to do instead? You're going to write it down. You're going to transcribe it. Transcribe it onto, I'm going to say paper recipe. Okay. Now, when we undergo transcription, that's exactly what's happening is we're just copying it word for word, letter by letter. But we went from the electronic format to a paper copy. So all we're doing from DNA is we're copying it into a different format, RNA. Okay. Now, what do you do with that paper recipe?
Make it.
You go make it. Yeah, exactly. You translate it into, I'm gonna say you take it to the grocery store and you translate it into ingredients. Now, do your ingredients look anything like your online recipe? It's a completely different format, right? You're not pulling a squash out of the computer. Okay, so that's the idea here of the central dogma. Okay. Hopefully that kind of helps solidify that a little bit there. Okay, so when you make a protein, understand your protein looks nothing like the RNA, looks nothing like your DNA. Okay. And who is responsible for turning your paper recipe into ingredients? Who's responsible for turning the RNA into a protein?
Right.
Ribosomes. Okay, so if you're keeping count now, that's 3 times I've mentioned ribosomes translating. Wink, wink, nudge, nudge. Okay, so hopefully you guys are seeing that my lecture style is pretty straightforward. Okay. All right, so this here is going to be like an assembly line. Okay, I'll just give you guys I'll give this one away here because it's kind of weird. This is the endoplasmic reticulum. Okay, with the endoplasmic reticulum, you have the smooth and the rough. Okay, now, anybody remember what does the smooth ER take care of? Lipids. I'm going to say lipid processing / assembly. Okay, what about your rough? Proteins. Now, this is going to be an example of an exam question here. So rough is processing / assembly. When it comes to your exam questions, some of them are superficial. Yeah, like your quizzes. I get burnt out from writing exam questions because they suck. But kind of what separates the grades is application questions. So I haven't talked about this yet. What gives the rough endoplasmic reticulum the ability to process proteins?
The rest is the rest.
So again, the question was, what gives the rough ER the ability to process proteins? Correct answer was ribosomes. How did you know it was the ribosomes? Yeah.
Down there,
Yeah, they're found there. Absolutely. What do the ribosomes produce?
proteins.
They produce proteins. Okay, so what I'm getting at is with the exam questions, it's kind of like in between the lines. It's about this application. You need to know that ribosomes makes proteins, but I just applied it to a different scenario here. Okay, so I think after these metaphors, I think it's more. I'll give you guys more examples. Okay. These are your actual worker bees. These are the ones actually making the proteins. Therefore, these would be ribosomes. That's why I was talking about the assembly line. You got to have workers on set assembly line. Okay. Ooh, talking about spelling. You can call me out, by the way, if my spelling is wrong. I will not be offended. Okay. All right, and this one here. Yeah. Cytoskeleton. Okay. All right, so let me think. Let me practice with names too. Meredith, got one. Pick an organelle, random one. Doesn't matter. Anyway, there's not a wrong answer, I promise. Anybody want to help her out? Random nucleus? Okay, let's do the nucleus. So an example quiz question could be, where does, where does replication happen inside the cell? Where does replication happen inside the cell? Nucleus, why? Contains DNA. Replication, what are we making? There you go. Okay. Ooh. Okay. When it comes to exam questions, really, as you guys are going through the material, you should be asking things like why. or how. Those are really kind of the big picture things. If you think about it, when you guys become nurses, patients are going to ask you these questions. Why is this happening to me? How did I get this disease? What do I get to look forward to? How's my life going to be changed by this? Okay, so let me think. I'm trying to think of an example that's not like too crazy. Okay. Let me just write this question down with the jargon, and I promise we'll break it down, okay? Developing zygote has normal nucleus. How will this affect development? Okay, so the question is, a developing zygote has an abnormal nucleus. How will this affect development? Okay, what the hell is a zygote? We didn't go over that. Doesn't actually matter to this question if you need to know that or not. Okay, because the question is really getting at, we have an abnormal nucleus. We have an abnormal nucleus. So what does abnormal usually mean? Not normal, impaired, messed up, right? Now, if the nucleus is impaired, how will this affect growth? So let's think about this. Your nucleus contains your DNA. Why do we need DNA? Yeah, to create cells. It's the instructions, right? You ever try to build IKEA furniture without the instructions? What's going to happen? Even with it, right? You get the Hagen Frozen dresser and it turns into a bookshelf. So if we have a nucleus that contains DNA, the instructions for life, and our instructions are wrong, what's going to happen? Yeah, I'm going to say like it impaired growth, right? Your DNA contains the instructions for life. If your DNA instructions are faulty, the development's going to be faulty. Okay, that's more of the application, kind of what I'm talking about here. Yeah, so, and I'll try to go through some more examples there. So take, for example, mitochondria, our plant of the cell. If a cell is missing mitochondria, what would this cause in a patient? Maybe some metabolic disorders. Why? But they have plenty of glucose. What's wrong? Can't burn it into ATP. So they might feel fatigued. They might feel tired. They might have those metabolic disorders, but we haven't gotten into any yet, so that's why I'm kind of avoiding that. But you guys see what I'm saying there? Asking how or why. How would this affect a patient? Why does this affect a patient? Those are the application questions. Those are the big questions I'm going to be going for. Okay. All right. So let's take a little break here before we jump into some more cellular stuff. Let's go for like 7 minutes. So 9:25, we're going to pick up lecture again. Okay. I don't pause recordings. So when you guys rewatch this, it'll be 7 minutes of like chit chat and weird. I don't pause it because I forget to resume it. So that's why it's just going to run. Okay. But yeah, let me know if you guys have any questions or anything like that. 9:25, we'll pick up and do a little bit more.
Do you want us to leave the classroom to like have a snack?
Whatever you want to do. You can hang, you can chill. You know, no murder. Murder's generally pretty bad.
So the Zoom lectures will be online as well.
So yeah, everything like, well, that's one of the things it took a little while to figure out, but one of the feedback that I always got was I wish there was a way to record lectures. So by me recording this, I can post it to YouTube, and when we have the online lectures, we'll just post it to YouTube right after that. So you'll have an entire archive. One thing students have done was they'll create like a Word document of all the lectures, like the YouTube links throughout the entire quarter. So that way you can rewatch that when you hit MedSurg 1 and you're talking about or actually, how about this? When you go into peds and you're talking about first trimester development, talking about zygotes, sometimes they'll hang on to those lecture links and just rewatch it. So yeah, you'll have an entire library if you want it.
Would you know how to do that? Show me how.
Yeah,
Put it onto the Word document?
absolutely.
Thank you.
Yeah,
I'm
absolutely.
not that technical savvy.
All good. Everyone has to start somewhere. So, yeah, more than happy to. I try to make it pretty seamless. I haven't deleted videos. Yeah, I haven't deleted any of my lecture videos. So. They're all set to unlisted though. So if you try to search for my lectures in YouTube, it won't show up. You need the link.
Oh, okay. So I can't put you on the TV. Or can I?
What kind of phone do you have? What kind of TV do you have? Do you have like,
I have a smart TV.
so you can cast it? So yeah, you pull it up on your phone and then you can cast it to your TV.
That's perfect.
Yeah, I felt bad because like back in the day, students used to have like the tape recorders, like the audio recorders, and just sit here and record it. And then like one student was telling me, yeah, I record all your lectures. I hear your voice more than my husband's. Starting to get awkward. Another one's like, yeah, I listen to you while I lift weights at the gym. Whatever gets that power plant going, I guess. But yeah, I try to make it pretty accessible. The reason why I don't like so I go a step further with the YouTube thing. Some professors just leave it at Zoom. They list all the Zoom links. You click on that, there's a password. The reason why I don't do Zoom is like, what if you're driving? What if you're somewhere that you don't have internet? Okay, perfect. So you can stream it. YouTube is just a more stable platform, I find. So that's why I prefer YouTube.
So on here, it says like the Zoom link on the slope, that you would just go to the part on Canvas where the link is.
So yeah, right at the home page there. And you just, I already embedded it. So you just click that and it's gonna jump right into my Zoom.
Oh, okay.
So as long as you're in there before 8:15, you're good.
So since tomorrow's pre-recorded, where do we find that?
Great question. I will put it in actually right now while I have time.
So we don't need to use that Zoom link tomorrow.
Tomorrow you do not need it. I'm gonna put that recorded lecture right here.
Okay.
Actually, I'll do it right now. Just give me a sec. So you can actually see that link. And I'll post today's lecture, hopefully this afternoon in there. It'll just be in between classes. If you don't, I mean, you're gonna see it, I would say I get done at 6:30 today, so 7 by the latest for today's lecture. The reason why I try to post the lectures pretty quickly is because your quiz is always on the previous lecture's material. So your quiz tomorrow on this stuff. Okay, then you'll watch the cancer lecture. Then when I see you again next week, your quiz will be on cancer. Okay, so it's not gonna be like cumulative quizzes, but some of the material does stack. And I'll try to emphasize that when we get there. But generally the quizzes are pretty focused on that previous lecture. Where the- oh, there it is. Nope. Uh-oh. Wonder if I didn't download it.
So then we'll all be together throughout the next 21 months.
Pretty
Is
much,
that kind of how it's-
pretty much. Yeah, so you guys will have your cohorts here, and generally with cohorts, you guys stay together. So take for example, when you guys are finished with quarter one, going to quarter two, you guys will all see each other for farm and so on and so forth.
Well, it's definitely the first
Sounds
thing.
awful.
I think the bulb exploded.
I think that's, yeah.
Uh-oh. Well, shit. No. Well, that sucks. Can't fix that. So, question is, what the hell is? Yeah. I just don't know. Let's see. So you might have to pull out your computers and jump up to that Zoom room so you can actually see what I'm writing down. You guys might have to share computers. I'm gonna email see if I can get a hold of the IT help guy. That has never happened to me.
Okay. He's out. Where you guys from? Where's home though?
That's here, but it was California, and
Okay.
it's South Carolina, and it was Illinois.
What part of California?
Northern.
Okay, like, Travis Air Force Base or something?
He was stationed there, stationed in South Carolina, yeah, he moved to California. Francis was there for me, yeah. So,
How's it feel?
yeah.
And then do you look around here?
He was in Wilmington.
Okay.
Terrible.
How long is it?
5 years.
Okay.
Or if he did one in last semester.
So we're doing,
Yeah, so if you guys jump into Canvas, go to that Zoom link.
Okay.
That way, and I'll keep sharing my screen here so you guys can actually see it close up. It's basically now, instead of that screen, you got your screens.
Okay.
That's the goal there.
I don't know. It is.
Recording in progress.
You might want to mute yourself.
Recording in progress.
Her name's Whiskey. Tess, maybe. Yeah, I'd say turn your sounds all the way down too, just in case. So far, it's good.
Recording in progress.
One more. What a day one. I know. Sorry, guys, for the technical. If this happens, I don't know. It's like I'm supposed to lecture in here too for another 3 hours, and I have to go like steal somebody else's classroom. Hopefully, it doesn't come to that. All right. So hopefully, you guys can see my screen and all that, though. Cool. All right. So difference between a prokaryote and a eukaryote. So again, a little bit of Latin here. Pro, so it's like a prologue. Where do you find a prologue in a book? The beginning, yeah. So pro means before. Karyote means nucleus. Okay. Eukaryote, EU translates to good / true. Karyote nucleus. So this is what I'm talking about is if you know a little bit of Latin, kind of already know what's the difference between these two. Perfect. So prokaryote. Prokaryote, no nucleus. So that's kind of like bacteria, viruses to a certain extent. Eukaryote, that's like you. Okay. Also, yes, you're going to be bombarded with dad jokes. Heads up. I think I'm funny, but just laugh to make me feel better, okay? So proteins and lipids in the membrane. So all of our cells are surrounded by a phospholipid bilayer. All of our cells are surrounded by this phospholipid bilayer. What does bi mean? Two. Yeah, double, two. So that means all of our cells are lined by 2 layers of fat. Okay. Why is this important? That those 2 layers, the lipids, they will keep stuff in / out. So by surrounding everything by lipids, your cells insides stay inside. The stuff on the outside stays outside. Okay. The other part of it is the proteins. Got to have some proteins to allow stuff to go in / out. Okay, so 2 parts. The lipids help separate things. The proteins allow things to go back and forth, either from outside to inside or inside to outside. Okay. And I'm going to give you a little bit more context about this here. Okay, so there are 2 ways to kind of bring things in and out of a cell, passive versus active. What does passive transport mean? Perfect. No ATP required. Therefore, active transport requires ATP. Pretty straightforward. Okay. So with passive transport, there are 2 types of passive transport. You have simple diffusion. So let me kind of put like a And you have facilitated diffusion. Okay. For simple diffusion, this is where things go from high concentration to low concentration. And this is going to go right through the membrane. Okay, so simple diffusion is something that can just phase right through that lipid bilayer. Okay. In order, so let's think about the things that are allowed to go right through your cell's membrane. They have to be small. They have to be uncharged and lipid soluble. Why do they have to be lipid soluble? Yeah, it's because your membranes are made of lipids. If you want something to go through a lipid, it's got to be lipid soluble. So, what's an example? What's something that's small, uncharged, and can go right through your lipids? Water's is going to be water soluble, so water's not lipid soluble. Give you a hint, you're doing it right now. Oxygen, CO2, gases. So, you breathing right now, how much energy does it take to bring it into a cell? None. How do you get oxygen into a cell? Yeah, passive transport just goes right through the membrane. Okay, so that's going to kind of play a role in MedSurg 1, especially with anesthesia when you guys start learning about that kind of stuff. Facilitated diffusion. This is where you have a specific protein channel. Specific protein channel. So this is when, with these protein channels, they only allow one thing through. Okay, you'll need a different protein channel for different things. So now it's going to be for the kind of the opposite stuff. So stuff that is large, charged, and now water soluble. Okay. So what's an example of something that is large or charged or water soluble? Sodium. So in order to get sodium into a cell, you need a specific protein channel. How much energy does it take to get sodium through that channel? None. Okay. What else?
Potassium?
Potassium. That's another one. Absolutely. In fact, we'll just do all of the electrolytes. So sodium, potassium, chloride, calcium. Okay. What about the stuff you burn for ATP? Would that be through simple diffusion or facilitated diffusion? Facilitated, because glucose is actually really large. Okay, so how much energy does it take to bring glucose into your mitochondria? Which makes sense. If your whole point of the mitochondria is to make energy, do you want it to cost you energy? No. Okay. The other thing here is water. Okay. Now, active transport, that's the one that requires ATP. We'll talk more about this towards exam 2. It's really the sodium. It's really the sodium potassium pump. Sorry, I didn't know it was locked. Yeah, the bulb is kind of like just hot. Yeah, I unplugged it because like it wasn't shutting down. So it just has a line for the power supply.
You want to see right now?
Yeah, if you want to, we're good. Yeah. Yeah, just because I'll have class at 11:30. Yeah. Cool. So really what we're going to be focusing on is the passive stuff, not so much the active stuff. Why do we care about this like nitty-gritty stuff? Pharmacology. when you guys start talking about meds, you start talking about like, take for example, ointments or pills. This is going to change how the body absorbs it and how long it stays in the body. Okay. So again, with the examples there, you guys heard. Sorry. Yeah, would that work? I'm sorry. Okay, so yeah, in terms of questions wise, like that's really what I'm gonna go for is how much does it take to bring into a cell? What is passive, what's facilitated? Okay. All right. So I do a lot of weird random fun facts in this class just because I'm a nerd. So this is actually a computer generated photo and it costs $5 million to make this photo. But it is the most accurate photo of a cell. All right, so at the very bottom here, what's that sucker? It's mitochondria, yeah. Okay. These are your, it's hard to kind of see the colors. That's your specific protein channel for facilitated diffusion. That is inside you millions and millions of times. Well, it's a lot of this stuff you don't even know what's going on because it doesn't cost any energy. That's weird. That's cool. Okay, different types of cellular signaling. Glenn will be your friend. Auto, if something is automatic, something is autonomous, what does auto mean? Self. Crin means signal. So if a cell secretes an autocrine signal, who's it talking to? So it's telling itself to work harder, do more. Okay. Paracrine. Actually, let me stick with blue here. Para is something that you're going to see pretty consistently. Para means like next to or neighbor. So if a cell secretes a paracrine signal, who's it talking to? The cell's in its close proximity. So you guys, we're in a class right now, right? I'm only talking to you guys. This is a paracrine signal. I'm not yelling to the people in the hall or upstairs. Okay. Hormonal. I don't have any land for that one. Sorry. Long distance. That will come up more towards the end of the quarter. Neurotransmitters. That's going to be neuron to neuron. And then neurohormonal is combining those. This is going to be long distance neurotransmitters. Okay. So to be completely frank, really, this is where we're living for exam 1, autocrine and paracrine signals. Hormonal, this is going to be more exam 6. You don't need to write this down. This is just giving you a roadmap. Neurotransmitters, that'll be exam 2. And neurohormonal, that's exam 6. Okay, again, you don't need to write that down, just giving you guys kind of a roadmap. Don't worry about 3, 4, and 5 right now. Okay. So here's some pictures there. Again, I'm a very visual person. So hormonal, we're taking signals and just dumping them into the blood. Easy enough. Paracrine, my neighbors. Autocrine, myself. Okay. All right. So going back to high school, what are the 2 types of cell division? All right, perfect. Oh yeah, is it? Oh, okay, no worries. We got mitosis and we got meiosis. Okay. Now, which one's, actually, let me rearrange this and give myself some room. Meiosis. Which one's asexual? So it's mitosis that's asexual. So the way I remember this here is I actually have a younger sister. Talking to my sis, right? Mitosis is not sexual. Fucking toe. It's disgusting. Okay. Should also warn you, day one, like I usually don't curse too much. I try to ease you guys into it, but cursing will happen. Okay. So mito, not sexual. It's gross. Meiosis. Anybody ever see Austin Powers? You get the whole goal there is to make mini me's. So that's the way I remember it. Okay, so with mitosis, I'm just kind of diagram this out. I promise to explain what these mean here. We start with a cell. It has two sets of DNA. I'm going to make two cells out of it. If it's completely asexual, how many sets of DNA should be in each cell? It's the same amount because I'm just copying it. It's asexual. So think of it like this as your skin cell. You start with one skin cell, you're going to make two new skin cells. These two skin cells are complete, intact, have two sets of DNA. Okay. Now, meiosis, this is the sperm and the eggs. This is where now we get the gametes, where each one of the swimmers or the eggs has one set of DNA. Okay. So eggs, 23 chromosomes. Sperm, 23 chromosomes. Your liver cell, 46 chromosomes. Okay. So this kind of brings up the next bit here is terminology stuff. Okay. We have chromosomes. I'm just going to abbreviate as chromos. Chromosomes, pairs, and sets. These are three different ways to count the genetic material inside of a cell. Three different ways to count it, okay? So let's start with sets because that's the easiest. You get one set from mom, one set from dad. So in you, the majority of your cells, how many sets do you have? Two. You get one from each parent. Okay, so I can say you have two sets of DNA. Each set has 23 chromosomes. So you got 23 chromosomes from Ma, 23 chromosomes from Pa. How many chromosomes in total do you have? 46. So you guys see how I can say you have 46 chromosomes or you have two sets of DNA. Same thing, just different ways of counting. The hard one is pairs. Okay, so when we draw out somebody's chromosomes, chromosome 1, 2, 3, 4, all the way to chromosome 23. That's from mom. 1, 2, 3, 4, all the way to 23. So you see how I have two chromosome 1s. That's a pair. So how many pairs do I have? 23. Okay, we're going to get more practice with this, but this is one of the things that can be tricky out of this first lecture. Okay, is I can refer to it as chromosomes, pairs, or sets. Okay, now this picture here, no, you do not need to memorize this. No, I'm not going to ask you what the hell telophase is. I don't know. Okay, all I care about is the end products. All I care about is the end product. So, recapping, mitosis. At the end of mitosis, I should have two cells. In each cell, how many sets of DNA are there? Two sets at the end of mitosis. How many pairs are in a cell at the end of mitosis? 23. How many chromosomes are in a cell at the end of mitosis? 46. Now, if we switch over to meiosis, how many chromosomes are in a cell at the end of meiosis? How many chromosomes are in a cell at the end of meiosis? So meiosis being sexual. Your eggs and your sperm,
It's 43.
23. 23 chromosomes. Okay. How many sets of DNA are in a cell at the end of meiosis? One set. Okay, so actually, let me kind of change this. Let me clarify this here so that way it's not crossing. So let me say terminology. Okay, at the end of mitosis, asexual, you get 46 chromos. You get 23 pairs and or 2 sets. Okay, now in meiosis, you're making eggs and sperm, it's half that stuff. So how many chromosomes at the end of meiosis? 23. How many sets? One set. How many pairs? So think about it. Look at your feet. How many pairs of shoes do you have? You have 1 pair of shoes on your feet. Agreed? You have 1 pair of shoes on your feet? I hope. Take your left shoe and throw it away. How many pairs do you have now? None. So you guys are going to find as students, the 2 hardest answers for students to produce are none, no, or nothing. Yeah, it's you're so ingrained when I ask you a question that there's a positive answer, right? I push you guys to think critically because as a nurse, you better be able to say no, nothing, and none. Okay, so those are viable answers too. And you're going to find it really goes against the grain. Okay. So again, I don't care about the phases. All I care about is the end products. Okay. This is a cool GIF here, but I'm just going to skip it. Tissues. What is a tissue? And please don't say Kleenex. I make the dad jokes around here. My material's a little better.
So group of cells.
Yeah. Similar cells with similar function. With similar function. So I write function a lot throughout this quarter. So you'll see me abbreviate function as FXN. That just means function. Okay. Now, sometimes, like I've been editing these slides over and over again, and each quarter I edit it. Sometimes I kind of forget a little bit here. So I might ask you a question. What are the 4 main tissue types? What I might forget is that the answers are on the next page. So if I ask you a question, just play along. It makes me feel a little better. Okay. So epithelium. What is epithelium? Skin. So I'm going to say skin / coverings. Okay. Now we're going to, just like the anatomists of old, we're going to skip connective tissue. Muscular. There are 3 types of muscles in your body. What are the 3 types? Cardiac, smooth, and I heard it. Yep. Skeletal. Very good. Nervous tissue. Really, it's just neurons. So i.e. brain, and I'm going to say nerves. Okay. So now we're going to go back to connective tissue. What's an example of connective tissue? I'm jumping right to it. Yeah. That's weird. What does blood hold together? How the hell is that connective tissue? I think that's what happened. If you're ever confused about what kind of tissue it is, it's connective tissue. They just threw it in there. So blood, I was thinking like tendons, ligaments, yeah. What about bones? The bones are connective tissue. Okay, so just heads up, connective tissue is kind of like a catch-all for anything that's not epithelial, nervous, or muscular. All right. So we are jumping into genetics here. So fun facts. You don't need to write down these fun facts. I'm not going to test you on it. Like I said, I'm a nerd. I love this stuff, and I really want you guys to have an appreciation for the human body. Like it is such a statistical probability that you are here right now. It's absolutely crazy. Okay, so one of my pastimes, yes, I work too much. One of my pastimes I like to do is I go through textbooks and try to figure out what's wrong with their pictures. What's wrong with this picture out of this textbook?
The chromosome.
Yeah. That DNA is just hanging out like that. That is not the case. Your DNA needs to be super coiled. You don't need to write any of this down. Your DNA needs to be super coiled, okay? And again, this is just a fun fact. You don't need to write this down. Within each cell, if you were to take out the DNA, you would have 6 feet of DNA. Now, I am 6 feet tall. I'm sitting down, so you have to trust me. From foot to head, if we stretch out that's how much DNA is in every single cell. You can't just have it hanging out like this. It's crazy, right? Imagine like a shoestring. You start twisting that shoestring. What happens to that shoestring? It coils. That's what happens to your DNA. Now, another fun fact. If I were to take the DNA out of all of your cells and stretch it end to end, do you think you could touch the sun? Now, keep in mind, it takes 8 minutes for light to reach Earth. That means if the sun exploded right now, we would not know for 8 minutes. Could you touch the sun?
Yes, sure.
200 times you can. That's inside of you right now. That's crazy, right? And all of that working together is what brought us here today. I'm a nerd. I like this stuff. All right. So all of your DNA is encoded in these 4 letters here. No, you do not need to know what they are. No, you don't need to worry about their parent. Don't care. Again, just have an appreciation for how amazing you are. Like imagine going home tonight, your homework, I want you to create life using 4 different Lego blocks. That's you. You have 4 different Lego blocks. That's it. And here you are. Isn't that crazy? Yeah, I don't know. I like it. I think it's pretty cool. All right. So just a little bit of terminology here. When we take 3 of these nucleotides and group them together, that's called a codon. So a codon is 3 nucleotides together. Those 3 give you an amino acid. You put a bunch of amino acids together, you get a protein. So just a little bit of terminology. Okay. So as you guys can see, the 3 nucleotides make a single codon. I'm just highlighting this right now. You don't need to write this down. Codon 7 is a stop codon. When we reach a stop codon, that means we're done. That's the end of the chain. We don't need to translate anymore. Okay. Now, this chart here, this chart is how you figure out which amino acid comes from the 3 different nucleotides. You're going to have to memorize this for exam 1. Okay, you guys took that way too easy.
You're lying. You're definitely lying.
Yeah. Should you trust anybody? Like, what? Don't trust me. Don't trust me. Okay, couple of things here. You guys are going to be nurses. You're going to have to think for yourselves. Do not blindly follow authority. Okay? I'm not generally about like going to lie to you, but you should all remember always like, no, nothing. Those are fine answers. Okay. Why would I have you memorize this?
Because you would, you're mean.
Have I come off really as that much of a dick already? Okay. This stuff here, you could Google it. That's not the point of why you're trying to become a nurse. That's not just nursing. It's not just information you can look up. Okay. Also, again, if you get into a conversation about nucleotides as a nurse, I will pay you $200. Okay, unless you want to get into like fertility treatments or things like that, like then that matters. But no, the whole point of this class is not for you just to memorize a bunch of little random things for you to dump. Okay, so no, not going to ask you to translate anything like this. Not even going to give you the picture or ask you to translate. I promise. Okay, what I want you to kind of see here is a certain theme. Where, take for example, if I do C U U C U C C U A C U G. What do you guys notice? What amino acid does that code for? Leucine. So what I'm getting at here is, this is why, one of the reasons why you are here today. You see how we have a different letter here for each one, but we still get the same amino acid. This is like foolproofing your stuff. So the actual term I'm getting at, this is called redundancy. Redundancy. This one you want to write down. Okay. Why? If your DNA is 6 feet long and you're copying this over and over again, are you going to make mistakes? Yeah, you're definitely going to make mistakes. Here we've made 4 different types of mistakes, but is it okay? Yeah, we still get the same amino acid. So what I'm getting at here is redundancy helps protect against errors. Okay, if I asked you to copy the dictionary down 3 letters at a time tonight, could you do it? I think so. I have faith in you. But if I want you to do it 100 million times, you're probably going to make a few mistakes, yes? Some mistakes can be bigger than others. And I'm highlighting this area right here. So I'm going to scroll back up. You guys don't need to write this down. But you see codon 7 is the stop codon, right? What if, let me say, what if codon 4 was misread as a stop codon? So what happens if we have a stop codon in the middle of this protein? It's not going to finish. So we will not translate the whole protein. How can this be a problem? How will this be a problem for a patient? There might be some deficits, there might be just some deformities, some impairments. Again, think about it. Your DNA is the instructions for life. If you're missing half the instructions, is that going to be a problem? Yeah. So that's not a question. Sorry. We will not be able to translate the whole protein. So I'm going to say there will be deficits / impairments in growth. Okay. So again, just recapping this, what protects us against these terrible mutations? Redundancy. So is redundancy helpful or harmful? Okay, perfect. Okay, so protein synthesis, we kind of talked a little bit about this already. Transcription, where we go from DNA to RNA. Anybody remember, where does transcription happen? Where do you find DNA? Nucleus. Okay. Now, another fun fact here. Who has a longer strand of DNA? You or an onion? Yeah. Onions have longer DNA strands than humans. Onions, their DNA strand is 30 times longer than ours. We've been to the moon. We have figured out so many things, and here we are, inferior to onions. What? I mean, yeah, like they're complicated, they got layers and all that, but what is going on there? It's because when you guys make copies of body cells, you need to understand what to keep and what not to keep. So this brings up introns and exons. This is kind of counterintuitive, so I'll explain it here. Okay, introns, this is going to be useless DNA. Exons are useful DNA. Okay. Onions are primarily introns. Yes, they have a longer DNA strand, but it's just this repeating nonsense. Okay. So when you guys are making a cell, let's just say you're making a new liver cell. We have a bunch of introns. Do we want to keep the introns or throw them out? Yeah. So introns are, I'm going to say, discarded. Exons, these are kept. Okay. Your liver doesn't need to remember how to be a heart. Your heart doesn't need to remember how to be a kidney, etc. That's why we only keep a small bit of DNA. Okay. So exons are the important ones that we want to keep. Translation. Where does that happen again? That's like 4 times now. Weird. Okay. So again, you don't need to memorize this picture. It's a whole lot of craziness that's happening inside of you right now. Okay. All right. Okay, let's see where we're at. Yeah, we'll do a little bit more and then we'll take a break, okay? All right, so a mutation, technically defined by your textbook as any inherited alteration of genetic material. Nobody talks like that. So just simply put a mutation, change in DNA. Okay, a change in your DNA. Now, are all changes bad?
No.
What protects us from a catastrophic change? What protects us from a catastrophic mutation? Redundancy. Okay. Now these are the different types of changes or different types of mutations that we can have. A point mutation, also known as a base pair substitution. These are the same thing. Okay. This is where we change a single nucleotide. We change a single nucleotide. Okay, so just one of the letters. Frame shift, there's 2 types of frame shift, either deletion or insertion. So kind of defining it, a frame shift is when we have I'm just going to define it and I promise to show you, but a shift in the reading frame. So let me just define it and I promise I'll give you some examples here. So a frame shift is a shift in the reading frame. Deletion, so how you would read this is a frame shift deletion mutation. This is a removal of 1 or 2 nucleotides. Okay. So deletion is removal of 1 or 2 nucleotides. Insertion, that's when you're adding 1 or 2 nucleotides. Okay. Inversion, exactly as it sounds, is just flipped. Your DNA, just like a textbook, needs to be read in a certain direction. So this is just when things are backwards. So let's get into some examples here so you guys can actually kind of see it at work here. So up top, that original sentence, the fat cat ate the wee rat. Notice this is a sentence, each word 3 letters long. 3 letters makes a codon. Very good. So in a point mutation or a base pair substitution, notice I have changed only 1 letter. Okay. The fat hat ate the wee rat. Can you still read that sentence? Okay. If you got a text from your mother saying the fat hat ate the wee rat, would you scratch your head? Yeah, I was just like, what are they talking about? Right? Like you can still read it, but it's still a little confusing, right? Some letters not so bad, some letters not so good. So an example here is a silent mutation. So a silent mutation, again, I change only 1 letter. If I change only 1 letter, that means it's a point mutation. So a silent mutation is a type of 8-point mutation. So let me say silent mutation. This is a type of point mutation. Okay. Now in a silent mutation, I've changed one letter. Instead of the hat, it says cat with a K. If you got a text from your mother saying the fat cat with a K ate the wee rat, would you know what's going on? So in a silent mutation, we change one nucleotide, but there is no change in the amino acid. Which means we still code for the same thing. You guys remember how we were talking about the 3 letters and we all coded for the same amino acid? That's what's happening here. So from the outside, looking at the protein, we would know nothing is wrong. This is the best kind of mutation. You change something, but the end product is still the same. Okay.
Could that be considered redundancy?
Yeah, absolutely. This is why one of the things that redundancy kind of causes is it increases our likelihood of silent mutations. Mhm. Now, insertion. Notice if I use the term insertion mutation by itself, if it just says just insertion, I have inserted a 3-letter word. So if I use insertion by itself, that means I'm adding 3 nucleotides, I'm adding a codon, I'm adding an amino acid. Does the sentence still make sense? Yeah. Deletion, I'm just removing a 3-letter word. Now, if I say frame shift insertion, I have added, in this case here, 1 letter. What happens to the rest of my sentence?
Can't read it at all.
You can't, it's complete noise. Okay, so what's worse, a frame shift insertion what's worse, a frame shift insertion or insertion? Frame shift insertion is worse. Okay, now inversion is completely backwards. Whole thing's messed up. Okay, now this is going to kind of a little bit of foreshadowing here for next lecture, but we're going to be talking about things that are physiological and things that are pathological. Physiological means, I hate using this term in healthcare, but normal, typical, expected, good. Patho, disease, abnormal, dysfunctional, bad. Okay, so of these here, of these mutations, what would be the most physiological mutation? Which, if you were to have a mutation right now, which one would you want? Which one would be least impacting towards you? Silent mutation. So I would say the silent mutation is the most most physiological mutation, or the least pathological. Do you guys see how that's the same thing there? Most physio or least patho? Okay. So what one would be the most pathological? So we have frame shift, we have inversion. Between these 2 camps, both of them are pretty bad, right? This is going to be nursing school in a nutshell. So even though your questions on your quizzes and your exams are going to be multiple choice, process of elimination might not help you because there's 2 correct answers right there. Frame shift insertion and inversion. Can we agree those are the 2 worst ones? In nursing school, I'm going to ask you which is the worst one. Which one's the best one? Okay. So let's just say you have a textbook. You open up the textbook and you see that it's upside down and you're in the index. What does that tell you?
Things wrong.
Yeah, it's upside down. You're starting in the back. You need to flip it. Okay. Let's just say again, we use the textbook in here. Week 6, we've been doing all of the reading. Week 6, you find out the other half of your textbook is in German. How useful is that to you? It's not. Here you are. You've invested 6 weeks of time and money because now you can't return it, only to find out that your textbook is useless. Which one has impacted you more? Finding out that your book is half German or that you started upside down? So frame shift insertion is the most pathological. Why? Because we invested time and resources at the start. Okay. Because we have invested time and resources and we got halfway before we found out it's absolute trash, it's absolute garbage. If you open up your textbook and you're in the index and it's upside down, you just flip it, right? If we hand DNA that's absolute junk right at the gate, we can just toss it and start again. Okay, so that's why frame shift is more pathological, because we've lost that time. We're not getting that time back. Okay. Right. So you're gonna hear me go a lot. When I go don't worry about it. Okay. All right. So we're gonna take a break here because we're gonna jump back into chromosomes. Let's take a pretty decent break here. So let's go get like 25, 10:25. We'll pick back up again. Okay. So just under 10 minutes. Some of you guys really believed me when I told you you had to memorize that chart.
I've had professors that often.
Mm-hmm.
Believe you.
Mm-hmm. Yeah, I've had that too. I think the big difference though, like the way I kind of frame it a little bit, undergraduate's undergraduate, that's kind of like say for academic discipline and all that. But here we're not preparing you for academic discipline. We're working for something more professional. So that's the context there. So that's just me also having a little bit of fun messing with you guys because you guys I love quarter one because you still have the fear of God in your eyes.
You get to the last one, you're like, oh.
Yeah, everyone about quarter five, I think that's when callousness and jadedness really kind of set in.
Sure.
This was.
So far doing okay, you guys, in terms of like information? I know it's a little bit of overload. It takes some time getting used to 3-hour lectures. Yeah, even with my graduate, like my master's program, we never had 3-hour lectures. So yeah, this is one hell of an accelerated program. Lindsay's not so bad though. I think health assessment is just 2.5 hours. Lindsay's a great teacher too. I can speak highly of her. She same thing with her as with me too. Like if you pay attention in class, you'll be fine. Lindsay does a really good job preparing you guys for the HESIs.
For the exams, I know you said you give like questions in class, but do you give like for notes or are there any other resources?
Great question. So in terms of reviews in class, I don't, just because we're not gonna have time. But office hours, if you want to meet with me for office hours, like, hey, I'm struggling with this topic, or can we do like a brief rundown or something like that, be more than happy to. With those kinds of office hours, sometimes I'll stack it. I will put multiple students into a single office hour just to kind of, it's easier to meet with 3 people for half an hour rather than like half an hour 3 times. But I will never make it more than 5. So yeah, as you guys get to know one another, you start forming study groups and things like that, be more than happy to meet with you guys to do anything you want for exam stuff. I try to give you some example questions throughout lecture, as you saw with the quizzes, and then this now I'm going to be shifting a little bit more towards the exam. I try not to leave you guys unprepared. Okay. All right, so we're going to pick this up here and finish her out. Okay. So talking about chromosomal numbers. So here, oh by God, if I haven't scared you yet, Latin is going to be the key. Okay, you need to know this Latin here because the words are very, very similar. Euploid. So you guys remember EU, good/true. Okay. Ploid, we're gonna write this out here. Ploid translates to set. Ploid translates to set. So euploid, good/true set. How many chromosomes make one set? Think about back to your meiosis there. One gamete, one egg, one sperm has one set of DNA. How many chromosomes are in an egg or in a sperm? You have to have 23 chromosomes to be equivalent to one set. Okay, so a euploid means you have chromosomes 1 through 23. You have a complete set. Haploid translates to one set. Diploid, what do you think that translates to? 2 sets. Okay. Now, somatic cells. So when something is somatic, that means kind of like normal functioning. So normal function. So your skin cells are diploids. Your liver, your heart, your eye, those are all somatic cells. Those are all diploids. So somatic cells and diploids are synonyms. They mean the same thing. Okay. Polyploid. What does poly mean? Many. Many up here. Okay, so how many is many? More than 2. More than 2. So anything greater than a diploid is considered a polyploid. So you might hear something like triploidy. What does that mean? 3 what? Sets. You might hear something called tetraploidy. Is that going to translate to? 4 sets. Okay. Now, you being a diploid, you have 2 sets. That's 6 feet of DNA. A triploid, 9 feet of DNA. Tetraploid, 12 feet of DNA. Do you think you have room for any extra DNA? No. So triploids and tetraploids, unfortunately, a zygote that is a triploid or a tetraploid will not mature. They will not come to full birth. They might end up in a stillbirth kind of situation there. So triploids, tetraploids, extremely rare, and unfortunately, they are not really sustainable, if that's the correct word there. Okay. So kind of let's get some practice here. So haploid, what did haploid translate to? One set. So the nice thing about here is if you know a little bit of Latin, because I can ask you how many chromosomes are in a haploid? How many pairs are in a haploid? Actually, sorry, let me scoot this down a little bit. Scoot this down. So how many chromosomes? How many pairs? How many sets of DNA are in a haploid? Again, if you know your Latin, it's a little bit easier. So how many sets we got? Okay, which means we have how many chromosomes? 23. How many pairs? So if you were to draw this out, like if you're not retaining this, it's okay. What I recommend is draw it out. Haploid, you know, is one set. How many chromosomes equal a set? 23. So if you were to draw out the chromosomes, this gamete, this haploid has 1, 2, 3, 4, 5, 6, all the way to 23. And then you can count it out. Okay. Now a diploid, 2 sets. If you were to draw this out, 2, 3, 4, 5, all the way to 23. 1, 2, 3, 4, 5, 3. So how many chromosomes? 46. How many pairs? So with pairs, you want to count vertically. Actually, let me change this color here. With pairs, you want to count vertically. That's a pair. So is that. A diploid has 23 pairs. You see what I mean by drawing it out? Can help you guys visually see it. Okay, now the last one. Let's go pink. Sets. Again, how many chromosomes do you need in a set? 23. So for sets, you count horizontally. Because a diploid translates to 2 sets. You guys see what I'm saying here? It might help to draw it out, and as you guys get faster with it, it becomes a lot easier. Okay, so if I were to draw out a triploid, 2, 3, 4, 5, 23. So that way you don't have to memorize these. I don't ever want you just to memorize stuff. Again, my memory sucks. I wouldn't want that on you guys. Okay. So we'll do this one together. Chromos, how many we got? So 23 plus 23 plus 23. 69.
69, sorry.
Oh, you're good. You're good. You're not going to have to, you won't need a calculator for the exam, I promise. Okay. How many pairs? So count the pairs. That's a pair. That's a pair. Yeah, it goes all the way to 23. Okay. And sets, how many you got? 3 sets. Okay. Now go ahead and try to do tetraploid by yourselves. I'm going to draw it out, and then I'll give you guys a couple of seconds here, and then we'll regroup. Okay. So try to figure out the chromos, pairs, and sets for the tetraploid. So let's start with the easy one. For a tetraploid, how many sets of DNA? 4 sets. How many chromosomes? 92. Now the weird one, pairs. 46, because you have a pair here. And you have a pair down here now. Okay, so going all the way through, that should be 46 pairs. Now, should you memorize this? No. Again, if you know the little bit of Latin, you can draw it out. Makes your life a lot easier. Okay. We're gonna still get a little bit more practice with this here too. So now we're going to be talking about aneuploidy. So before we had euploidy, good true set, and euploidy, anytime you see a or an, that means not or no. So this is not true sets. Okay. So it's a cell that does not contain a multiple of 23. Instead, they might have 24, 22, just not 23. Okay. Case in point, trisomy. What's the difference between trisomy and triploidy?
Trisomy, it's like one
Mhm.
extra, that's like the 13.
The chromosome.
3 13s instead of.
Mhm. Versus ploidy. So this is what I'm saying is pay attention to the wording here. If you're reading very quickly on the quiz, you're like, oh yeah, triploidy, 3 sets, done. Might be talking about something else. Be careful between trisomy, triploidy. Okay. Somy translates to chromosome. Ploid meant set. Here we're talking about a chromosome. So trisomy, 3 chromosomes. Take, for example, trisomy 21. That means they have 3 chromosome 21s. Okay. You guys have probably heard of this disease. Trisomy 21 is Down syndrome. Now, Down syndrome, this is the most common trisomy. Sorry, I kind of ran out of room. So Down syndrome is the most common trisomy. Okay. You'll see me, if I put X's next to stuff like that, don't need to worry about that. Don't need to worry about that. So you don't need to worry about the first or the third point. Really, it's the second point that I'm getting at. Okay. So trisomy 21 is Down syndrome, most common trisomy. On the other side of that, you could be missing a chromosome, monosomy. What does that translate to? 1 chromosome. 1 chromo. Okay. And you do not need to worry about that subpoint. Now, how on earth does somebody get an extra chromosome? How does somebody lose a chromosome? This is non-disjunction. Non-disjunction is going to be an error in dividing genetic material. So non-disjunction error is an error dividing genetic material during meiosis. So what I mean to incorporate this, how does a person get trisomy 21? Yeah, this non-disjunction error. How does somebody get monosomy? Non-disjunction error. Okay. So this is what it kind of looks like. Again, you don't need to memorize this. So up top here, we have a cell with 2 chromosomes. It should be evenly split, 1 and 1, yes? If we have a non-disjunction error, 1 gets 2, another one gets 0. We did not split it equally, and boom, we have trisomy monosomy. Okay, so again, all I'm saying is take away non-disjunction is an unequal splitting of the material that leads to trisomy or monosomy. Okay, and what is the most common trisomy? Trisomy 21 or Down syndrome. Very good. Okay, so practicing this with this a little bit. Trisomy. Name? Haley, sorry. Haley, give me a random number between 1 and 23. 20, perfect. So let's say somebody has trisomy 20. Somebody has trisomy 20. So what does that look like? 1, 2, 3, 4, 5, 20, 21, 22, 23. 22, 23. Trisomy 20 means they have 3 chromosome 20s. Do you see how that's different from triploidy, where we had 3 sets? Okay, so same questions as before. How many chromosomes does a person with trisomy have? How many pairs do they have? And how many sets? Okay, give you guys a couple of seconds to figure that out. Okay, remember, you only count it if it's a complete set or a complete pair. So, how many chromosomes does somebody with trisomy 20 have? I get 47. Now, does that matter if it's trisomy 12, trisomy 21, trisomy 23? Same numbers. It's just different chromosomes that they have a third of. Pairs, how many complete pairs did you guys count? 40, ooh, sorry. 23, yeah. 23 complete pairs. How many complete sets? 2. Awesome. Okay. Monosomy, again, they're missing a chromosome. I'm going to steal this one here and just let's do monosomy X, for example. So what that looks like. 1, 2, 3, 4, 5 to 23. So 22, 23. 1, 2, 3, 4, 5, 22. Blank. Okay, so same exercise here. Give you guys a couple seconds. I want to know the chromosome count, the pairs, and the sets. Give you guys a couple seconds for that. How are we feeling? How about chromosomes? What'd you guys get? 45. Because we're missing one. Typical person has 46, monosomy, missing one, so it's 45. How many, let's go sets. How many complete sets do we have? 1 set, just because this one's intact. 1 set. How many pairs? 22. So you're going to hear me kind of advocate this all quarter long. Don't just memorize things for the sake of memorizing things. If you learn how to get to some point, you'll never be lost. Here, do I want you to memorize exactly how many pairs are in a trisomy patient? You should at least know how to figure it out. Okay. Again, learn the process rather than just the destination. Okay. So these are some examples of aneuploidy. So these are trisomies 13. So let me kind of be frank with you here, just because they do come up probably, I think they're going to come up in quarter 5 with peds and OB. I can refer to it as trisomy 13 or Patau syndrome. So heads up, on your quiz, on your exam, I can ask, patient has Patau syndrome. How many chromosomes do they have? How many pairs does a patient with trisomy 13 have? So know both ways, trisomy 13 or Patau. Okay. Same thing with trisomy 18 and Edwards. You do not need to know the stats underneath them. Again, just understand they are trisomies. Okay. Trisomy 21, Down syndrome, you guys already knew. Again, you do not need to know the stats here, but I do want you to take away this main correlation here. Okay. The number one risk factor is maternal age. Okay. The number one risk factor for Down syndrome is the mother's age. So case in point, if you guys look, the ages right there, that's the mother's age. The older she gets, what happens to the likelihood of giving birth to a child with Down syndrome? Increases. This is probability. This is not 100%. Okay, and to be honest with you, my parents, they had me a little bit later. My mother was about, I want to say like 43 when she had me. Don't have Down syndrome. I don't think so. I don't know. I never asked, never came up with the physical. So just because someone who's older birth to a child with Down syndrome, or even younger, right? If they give birth to someone like at a younger age, it still has risk for Down syndrome. What I'm trying to get you guys to take away from this is when you guys become nurses and you're counseling patients, what's my risk factor? What's the number one thing for giving birth to a child with Down syndrome? Maternal age. Syndrome and not Patau syndrome. So take for example, if somebody's gonna have trisomy. Sorry, my internet got wonky. If somebody's gonna have trisomy, why is it more likely, or why is it Patau syndrome? Oh, I'm sorry, that was a terrible question. All I'm trying to say is the most common trisomy is, that's the takeaway point here. Sorry. Okay, so if someone's gonna have trisomy, it's most likely gonna be Downs. And the number one risk factor for Downs is maternal age. That's what you need to worry about. Okay. All right, so here are some, this is some reading for Down syndrome. I will not test you on this. So if you want to put a line through it, go for it. But I know in health assessment, Lindsay does talk about this. Again, I won't test you on it, but this might give you some nice background for health assessment. Okay. All right, other examples of aneuploidy. So trisomy X. Trisomy X, what's going on there? Patient has trisomy X, what does that mean? If you look at their genotype, yeah, they're wild, 3 X's, right? What's kind of interesting about trisomy X is so let me X out the subpoint here. Is on the slide. You don't need to worry about that subpoint. I'm going to summarize it here. It is asymptomatic. What does asymptomatic mean? So A means without, symptomatic, symptoms, without symptoms, there are no symptoms, right? So would somebody know if they had trisomy X? No. The only time that a person finds out that they have trisomy X is if they get genetically tested or can't get pregnant. So infertility. So a lot of times women who are unable to get pregnant and they seek fertility treatments, they go for genetic screening and it can come back as trisomy X. I have had this genetic condition for my entire life and I never knew about it because it's asymptomatic. Okay. I think it says in there, it's like 0.1%. So it happens more often than not. Okay. So it's kind of wild. If you have an extra X chromosome, the only thing that like you're still going to go through puberty, you're still going to have menstruation, but you just can't get pregnant. Go figure. Okay. Monosomy X, also known as Turner syndrome. So that is X blank. Okay. So 45 chromosomes here, one set, 22 pairs, etc. Okay. Again, you do not need to worry about that second subpoint there. I'm going to briefly summarize this. This happens kind of on a spectrum, some kind of blanket statement, meaning providing a blanket statement here. Mild, physical / mental deformity. Okay, it's relatively mild. You don't need to take notes on this. It's just weird. But if you were to have, so ladies, XX, right? Would you rather have an extra X chromosome or be missing one? Isn't that freaking weird to have a little extra one? You would never know, but you're missing one. Oh, you definitely know, right? Speaking of sex here, we're talking about sex in terms of just from a genetic standpoint. Okay, which chromosome determines sex? The Y chromosome. So Y determines sex. Okay, so just because these terms kind of get used a lot in modern day language. Sex and gender, two different things. Sex is looking at it from a biological perspective. Gender is looking at it from a societal perspective. I am not poking gender with a 9-foot pole here. We're just talking about the genetic side, okay? So trisomy X would be what sex? Female. Monosomy X or Turner? Female. Now we cross the line into Klinefelters, which is XXY. Therefore, Klinefelters, by this definition, would be male by sex, or sex is male. Okay. With Klinefelters, this is severe physical / mental deformity. So a little bit of recap here. How does a person get Klinefelters? How does this person get an extra chromosome? How does somebody get trisomy? Non-disjunction error, okay? Non-disjunction error. Yeah, it's freaking weird. Our chromosomes are weird. So modes of inheritance, how do we get the different things here? Autosomal recessive. So you guys remember your little Punnett squares, the Mendelian beans, right? So like autosomal recessives, little r, for example. This is going to be the most common for genetic diseases. So take, for example, like sickle cell anemia. Okay. Autosomal dominant, they really don't exist anymore. So think about like if it was autosomal dominant, like take, for example, if it was dominant to be born with one leg, evolution would have weeded that out millions of years ago. We do not see really autosomal dominant diseases. Okay. X-linked recessive. So this is going to be, let me say, mainly affects males. Mainly affects males. Now here's your exam question. Why?
Y-linked trait.
So for males, it's XY. Females, you guys have XX, right?
It's the excellent recessive is because the other X makes up for it, right?
So examples here. Baldness / colorblindness. So who is more likely to go bald, males or females? Yeah. I'm losing my hair, it's like thinning. This is just a facade. I sit this far away for social distancing, and the fact you can't tell, I just Sharpied it in this morning. Okay. Why am I more likely to go bald? I only have one X. So whatever X I get, that's the one I have. Ladies, if you have the balding gene, that's okay, because you have a second backup set of instructions. Okay, can women bald though?
Yeah.
What happens if they bald? Well, you know, both X's are carrying that gene, that balding gene. Okay, so that's why X-linked mainly comes out in males, not so much females. Okay, thanks, Mom. Same thing with X-linked dominant, don't really exist. Okay. All right, so talk a little bit about genetics. It's a huge thing, so just keeping it very general. The genotype is just a set of genes. The phenotype is the observable attribute. Okay, so let me illustrate the difference here. A phenotype, so this is something that you can observe. It is dependent on three things. You need to have the genotype. You need to have the genes. They need to be activated. So I'm going to say activated genes. And the third thing is you need an environmental trigger. So I'll give you an example here. I have a gambling addiction. True story. Okay, I have the genes for a gambling addiction. I sure as hell know they're turned on. But I have never set foot in a casino. So do I express the phenotype for a gambling addiction? No, this is exactly why I don't gamble though. I got plenty of other addictions to worry about. Even though I have the genes, even though the genes are turned on, I don't display the attribute because again, I don't gamble, right? Another example, I believe it was in the 80s, there was a female athlete at the Olympics competing for long jump, okay? I believe she was competing for the US. She jumps the furthest, gets the gold medal. They drug test her. Her drug test comes back completely negative. She is clean. They still strip her of her gold medal. Because in her drug test, her chromosomes came back XY. She has XY. So she has the genotype for being a male. But that Y chromosome never activated. She matured as a female, went through puberty, menstruation, female characteristics, development, never knew she had the Y chromosome. Even though she has the genotype, that Y was never activated. So you see how you need all three things in order to see the observable trait. Okay. Yeah, this is like way back before the discussion of gender. So that's just the example there. If you want to observe something, okay? Slide here, of course, it's much more complicated. It's not just one gene controlled balding, it's several, okay? All right, so when it comes to studying at home, through the notes, you could re-listen to this, you could reread it again, but let's be honest here. You guys are at this point here, you guys know how to read. I do not recommend you guys just sit and read for 8 hours. You're proving nothing, okay? The best thing to do and the most time efficient thing to do is as active as you can with your notes. What I suggest, rewriting things. Don't copy, you're not a machine, you're not a parrot. Organization, put it in your own words. Make your own diagrams, make your own drawings, okay? So take, for example, chromosome pairs and sets. What I recommend, create a table. Go through all of the things that we talked about today. I'm not going to fill this out completely, but this is just to give you guys an idea. So the names of stuff here. So let's say diploid. You guys shout things out to me. What other genetic disorders have we talked about or names? Huh?
Trisomy
Which one?
21.
Trisomy 21. What else? I think I heard Klinefelters. What else? Monosomy. What else? How about a somatic cell? How about a haploid? What about Edwards? Okay, so you're going to go through that lecture. You can organize it however you want. What did I keep asking you for each one? Chromosomes. Yep, pairs. Sets. You can make your own table here. Do you see how this is basically, it's not asking you to copy your notes, but this is getting you to create your own diagram. Makes your notes a little bit easier, faster to read, makes you go through it again. Again, try to be as active with your notes as possible. Don't just sit there and read. You guys know how to read. Okay. Your time. Okay. Now you guys have a quiz on this tomorrow. I have internet. Okay. I might not know. I might not though. So if you come up with a table and you write it down on a piece of paper, feel free to text me a picture. Can you look over this? Can you make sure my counts are good? Be more than happy to. Okay. However, again, I'll be lecturing until 6:30 today. So it might be a little bit later. Okay, but I'd be more than happy to help you guys out here. I don't even know if I'll be able to check my email, but I'll try. Okay, but that's what I have for you guys. So good job on day one. Sorry about the lamp exploding.
Oh,